Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about Archaebacteria, their habitat, characteristic features, classification as well as applications. Let's start. At first, Archaea is a separate domain of life but with bacteria and eukarya containing single cell prokaryotes other than bacteria. So we have three domain classification, three domain of life, which was proposed by Carl Woese. So in that classification, which was proposed by Carl Woese, eukarya, archaea and bacteria is present. So archaea as a separate domain of life is present together with bacteria and eukarya. And these archaea are the oldest life form known to exist on earth. These are very primitive, ancient, with very simple characteristic features. The name archaea was derived from a Greek word archaeos, and archaeos means in Greek primitive. So they show primitive living characteristic like very simple structure, simple mode of nutrition acquisition, biochemistry, and they can survive in very extreme habitat, in extreme cold condition, in extreme hot condition, in extreme saline condition. So it is a very important and unique characteristic feature of Archaebacteria. They can survive in extreme condition. They are extremophiles. Next, initially under two kingdom system, archaea were considered as a type of bacteria and included under prokaryota domain and were known by the name of Archaebacteria. So initially bacteria and archaea together are included under a same domain known as prokaryota. But later the biochemical and genetic studies revealed that archaea have different metabolic characteristic and genetic constituent than that of bacteria. So in 1977, Carl Woese and George E. Fox has suggested archaea is different from bacteria based on their rRNA gene analysis and suggested that they are different. They are a different domain of life. So rigorous research and genetic and biochemical studies revealed that the 16S rRNA ribosomal RNA gene analysis suggested that the archaea is quite different from bacteria. So that group is completely different from bacteria, and they kept separately under a separate domain, that is Archaebacteria, archaea. So these are microscopic single celled organism, lack nuclei because these are prokaryotes, and they also lack other membrane bound organelle. Structurally these are similar to bacteria in their shape or size. That means archaea can be bacillus shape, of cocci shape, of oval shape. However, some species are flattened also. Some are square shaped like Haloquadratum walsibi and may reach up to 200 micrometer or more. So like bacteria, archaea are also aerobic. Sometimes these are anaerobic. They may be facultative or obligate. So they vary in their oxygen requirement due to such structural resemblance, but they have some structural resemblance with bacteria. So initially they are thought to as bacteria, but additionally additional to this bacterial characteristic, archaea also possess some eukaryotic characteristic. So they show some genetic and metabolic characteristics which are similar to those of eukaryotes. So eukaryotes have some similarities with bacteria. They have also some similarities with eukaryotes. So but it is different from those two. Next, ultrastructure of archaebacterial cell. Archaea is a type of prokaryote, but they have rigid cell walls and that are made up of various organic compounds like S layer. One is called S layer, which is proteinaceous layer and which contains glycoproteins. Then pseudopolysaccharides, which is similar to bacterial polysaccharides, but in some aspect the archaebacterial cell wall is different as they lack D amino acid and N-acetylmuramic acid, which is present in your bacterial cell wall. In bacterial cell wall, the bacterial cell wall is the chemical composition of bacterial cell wall is peptidoglycan, and which is the composition of peptidoglycan is N-acetylmuramic acid and N-acetylglucosamine. But these archaebacteria lack N-acetylmuramic acid and also lack D amino acids, which is present in bacterial cell wall. Then this archaebacteria contains pseudomurine, a layer found in methanogen that's similar to peptidoglycan but not peptidoglycan. Then it contains methanochondroitin, a component of cell wall. Then glutaminylglycan, a component of cell wall. Sulfated heteropolysaccharides, it is also a component of cell wall of archaebacteria. Then protein sheath, a structure that can support S layer. But here I must tell you the cell wall pattern of different archaebacteria is different. That means they show different types of cell walls. That means one archaebacteria so this type of cell wall, another so another type of cell wall. Cell wall composition, the pattern of arrangement differ in various archaebacteria. So this diagram shows schematic representation of cell wall. At first, A diagram shows a typical gram-positive archaebacterium, Methanobacterium homicucum. So they show this type of cell wall. They show cell wall, then cell membrane, then cytoplasm. But in case of a typical gram-negative archaebacterium like Thermoproteus tenax, they show surface layer, then cell membrane, then cytoplasm. So it is basic diagrammatic representation of gram-positive archaebacterium and gram-negative archaebacterium. Okay. These are the types of archaebacterial cell wall. So different species of archaebacteria, so different types of different patterns of their wall arrangement. Like this is Methanothermus, which shows at first S layer, then pseudomurine, then plasma membrane. Then Methanospira first contains pseudomurine, then plasma membrane. Then Methanosarcina contains methanochondroitin, then S layer, then plasma membrane. It is Sulfobels, which contains S layer, then plasma membrane. Then Methanospirillum at first contains sheath, then S layer, then plasma membrane. So they show difference in their wall arrangement, cell wall arrangement. So the cell wall of archaebacteria protects the cell from environment and involved in many functions, like it maintains the shape of the cell, it protects the cell against virus, heat, acidity, alkalinity, then it helps in the transportation. Archaea cell walls differ from bacterial cell wall in their chemical composition and lack peptidoglycan. Then the cell membrane and specialized membrane proteins. The cytoplasmic membrane of archaea is different from eukarya and bacteria. Their cell membrane is composed of glycerol ether lipids, unlike glycerol ester lipids of other domains. The phospholipid backbone of archaeal cell membrane is stereochemically different than those of eukarya and bacteria. So these archaea have SN glycerol 1-phosphate backbone, whereas other have SN glycerol 3-phosphate. And fatty acids in the membranes are structurally different in sense of that archaeal membrane fatty acids are long isoprenoid and highly branched, with multiple side chains, and some also contain rings of cyclopropane or cyclohexane, in contrast to bacterial and eukaryotic membrane fatty acids, which are straight without any branching and rings. So the fatty acids present in archaeal membrane are highly branched and contains isoprenoid. The membranes also house specialized protein called rhodopsins that function in capturing light and generation of proton gradient. So let's have a look on the structure of the cell membrane. So this is the structure, and the difference in how lipids are made up of in bacteria and eukarya compared to archaea is known as lipid divide. So that difference between archaea and other domain is called as lipid divide. So as I have already told you that bacteria and eukaryotes have glycerol 3-phosphate, SN glycerol 3-phosphate and fatty acid is linked to SN glycerol 3-phosphate by ester bond, but here we have glycerol 1-phosphate and ether bond. This is the difference between other domain and archaea, and this is the structure of the archaeal cell membrane. Okay. Another characteristic feature of this Archaebacteria is the presence of hammers. It is a unique cell appendage found in Archaebacterial cell, actually found on the surface of Archaebacterial cell, that helps the cell to attach on the surface and to each other. Hammers is long helical tube with barbed grappling hook at the end. At the end it have a barbed grappling hook, that means pointed hook. The hammers helps Archaebacteria to attach on the surface and to attach with each other, which may help to form biofilm and communities. What is biofilm? Sometimes in the environment we have some pollutants like plastic, like other pollutants. On that pollutant, bacteria at first colonize. So they colonize, that colony formation is called biofilm formation, and that biofilm formation is aided by the presence of these hammers. The hammers helps to form the biofilm on any pollutant. Then each archaebacterial cell has about 100 hemi, approximately 100 hemi. Then each hammers is 1 to 3 micrometer long and 7 to 8 nanometer in diameter. And the location hemi are found on the Archaebacteria in cold sulfidic spring. So the Archaebacteria which is present in cold condition and in sulfidic springs, that means springs, hot springs which contains mainly sulfur, in that Archaebacteria hammers is present. Then the genetics. Genome of archaea have a unique gene which codes for protein characteristic to them only. So they code for specific proteins which are present in Archaebacteria. Archaea generally have singular circular chromosome, a piece of circular double-stranded DNA located in the area of the cell called nucleoid. Archaebacteria also lack nucleus, a definite nucleus as bacteria, and the central position where DNA is present is called nucleoid. Their rRNA sequence and tRNA gene are also unique in many ways. Some archaeal gene may contain introns, which is a characteristic features of your eukaryotes. Horizontal gene transfer is common in many archaeal genera. The complexity of RNA polymerase. RNA polymerase of archaea are different and complex than bacteria. They are more closely related to eukaryotic RNA polymerase. Okay. Next, metabolism. Different species of archaea are capable of methanogenesis. What is methanogenesis? It is a process of production of methane gas during metabolism, and it is seen only in archaea. It is specific characteristic feature of archaea. Besides, these are capable of chemolithotrophy, photolithotrophy, hyperthermophilic growth. Then extreme habitat, I have already told you in introduction section that these are extremophiles. They can tolerate extreme condition, hypersaline condition, hyperacidic condition, hyperbasic condition. So these are extremophiles as they have unique cell membrane composition, chemical composition of cell membrane. Then classification, based on the phylogenetic relationship, archaea are classified into different phyla, some of which are well recognized and some are recently discovered and still proposed phyla. Some of them are introduced below, like first one is Archaeota. Archaeota contains containing diverse genera that can survive in extreme alkaline, extreme saline, and extreme higher temperature condition. That means they are thermophilic. Then they have Thermoproteota, which are mainly marine archaea, and then Nitrosospirota, then Nanoarchaeota, then Cora Archaeota, then Archaeota. So the pronunciation is very complicated, so please tolerate because it is very tricky. But once you remember this, you will not face any difficulty in pronunciation. Okay, so one is Cora Archaeota, then Archaeota. Next, ecological significances of archaea. The point number one and the most important point is they are capable of doing methanogenesis to produce methane gas. Then nutrient cycling in extreme habitats, as they can tolerate extreme conditions, they can perform the nutrient cycling there. Next is they are the part of biogeochemical cycling. Then they can also live as by performing the symbiosis or symbiotic relationship, by developing symbiotic relationship. Then they help to maintain the marine ecosystem. Then they have some human applications. So at first the enzymes of extremophiles can be used in industrial applications. They can be used to produce methane gas. Then bioremediation, they help in bioremediation. What is bioremediation? Remediation or cleanup of environmental pollutant using microbes. So by using this Archaebacteria, you can clean your environment. Then archaea are also used in fermentation process for the production of cheese and other fermented food. They are also used as probiotics. Some biorefinery processes involving the production of amino acids, enzymes, and biofuels are under study. Some Archaebacteria are found to produce polyhydroxy alkanoids, PHAs, which can be used to make bioplastics, which can be a best alternative of these synthetic polymers or your polyester. So bioplastics are eco-friendly in nature and biodegradable. They cannot make any pollution in the environment. So these are all the human applications of Archaebacteria. So it is all about Archaebacteria. I have collected data from different sources, from YouTube, from different links of Google, from book that I have. So please like it, please appreciate my hard work, share with your other friends, and subscribe to my channel Sunanda's Tutorial.